without and mixis means mixing. So here what we are trying to imply is that here seeds will be developed but without mixing, means without gametic fusion. Alright, so seeds are developed without gametic fusion, so that itself will be known as apomixis. Now apomixis is hence also termed as a type of asexual mode of reproduction which mimics sexual reproduction. Alright, so it's a process of asexual reproduction which mimics sexual reproduction. Alright, so now let us discuss a few types of apomixis. There are a few types of apomixis that we are going to discuss. First, Adventine embryony, also known as sporophytic budding. Adventine embryony or sporophytic budding. Now if you remember, the structure of the ovule, see this being the embryo sac, isn't it? So this is the egg surrounded by the two synergids, and when the egg will fertilize by the help of a male gamete, finally the fertilization will take place successfully, giving rise to a zygote, isn't it? And then an embryo will develop, right? That is going to be the product of sexual reproduction. See, about apomixis I have already told you that here seeds are developed without fertilization. Means embryo is going to be formed without any gametic fusion. Okay. So see, this being suppose a cell of the nucellus, right? And this being a cell of the integument. Both are diploid cells. Now if these cells, they start to divide and give rise to diploid embryo. In that case, this kind of embryo development from the cell of the nucellus or from the cell of the integument would be termed as Adventine embryony. Okay? So here, diploid cells of nucellus or integument will generate embryo. Alright. So this type of embryo development from the cell of the nucellus or integument would be termed as Adventine embryony or sporophytic budding. It is very commonly seen in citrus or even in mango. Alright. Next is recurrent agamospermy. Now again look at the diagram of the ovule. Okay. So if you remember the formation of the ovule or say megasporogenesis, what I had told you that first one of the cell of the nucellus which is nearby to the micropyle, it will give rise to archisporial cell, right? And then eventually the archisporial cell will give rise to primary parietal cell and primary sporogenous cell, right? The primary sporogenous cell will directly behave as megaspore mother cell, isn't it? This is the starting of the embryo sac formation, isn't it? So in that case, now suppose after the megaspore mother cell is formed, first thing that will happen is meiosis, right? So the megaspore mother cell which is diploid will undergo a meiosis to give rise to haploid megaspore tetrads, out of which 3 will degenerate and only one towards the salazar will survive, and that will give rise to haploid embryo sac, right? So now if I tell you that when the megaspore mother cell is formed, it does not undergo meiosis. Rather, it directly gives rise to an embryo. So what will happen? The embryo sac will be diploid, isn't it? Suppose this is a anatropous ovule, okay, and nearby to the micropylar tip, this is the megaspore mother cell that has been formed. So this is the megaspore mother cell. Okay, now megaspore mother cell, this is the megaspore mother cell. Which is of course 2n, isn't it? So megaspore mother cell expands and undergoes one round of mitosis giving rise to 2 nuclei, salazar end, micropylar end. Then the 2 nuclei, they move towards opposite poles, and being in the opposite pole, another 2 rounds of mitosis occurs. So 4 nuclei are present at opposite ends, isn't it? Now, from each of the pole, 1 nuclei shifts at the center. So from here 1 nuclei comes, from here 1 nuclei comes. Salazar end, micropylar end. So these 3 cells will now, or these 3 nuclei will get surrounded by thin peripheral cytoplasm, giving rise to diploid antipodals. And these 3 nuclei will also do the same, giving rise to a diploid egg apparatus. Isn't it? So here, the egg that is present in this diploid embryo sac, the egg that is present, that is haploid or diploid? It is diploid, right? So this diploid egg, when directly gives rise to an embryo, that would be termed as recurrent agamospermy. Is that clear? So in that case, do remember, in recurrent agamospermy, the megaspore mother cell directly will form a embryo sac, and of course then the embryo sac will be diploid. Why? Because megaspore mother cell has not undergone meiosis here to give rise to haploid cells. So the embryo sac is diploid, and it consists of a diploid egg. The diploid egg directly forms a diploid embryo, and of course then there is no fertilization here, rather directly a asexual mode, an embryo has been developed. So that's what is recurrent agamospermy. This is very common in apple. Alright. Now let me tell you about a third type, that's called non-recurrent agamospermy. Non-recurrent agamospermy is similar to recurrent agamospermy. Just a simple difference is there. Here in recurrent agamospermy, it is the diploid egg, right? The diploid egg is giving rise to diploid embryo, right? But in non-recurrent agamospermy, the megaspore mother cell will undergo meiosis also, and it will give rise to a normal haploid embryo sac also. So in that normal haploid embryo sac, the haploid egg that is present, that egg when directly gives rise to an embryo, that embryo is going to be haploid. And this type of haploid embryo formation is going to be known as non-recurrent agamospermy. So the only difference is that here in recurrent agamospermy, a diploid embryo is generated from a diploid egg, and here in non-recurrent, a haploid embryo is going to be generated from the haploid egg. That's it. There is no other difference. Okay? And that's why here in non-recurrent egg anomospermy, that means what is happening? The egg, normal haploid egg without fertilization is directly giving rise to embryo. And that we have learned so far has been called as parthenogenesis. Isn't it? This is what is known as parthenogenesis. A process in which without fertilization the egg will directly gives rise to a embryo. This will be again common in banana. Parthenogenesis after all. Isn't it? So that is all about apomixis. Now let us learn about another important term, that is polyembryony. Poly means multiple and embryony means formation of embryo. So see, usually a single seed is supposed to possess a single embryo, right? But in case a single seed consists of more than one embryo, we will call it polyembryony. So the meaning here is that presence of multiple embryo inside a single seed, that's what is known as polyembryony. Okay. Now, polyembryony was first reported by Leeuwenhoek in citrus. It was first reported by Leeuwenhoek in citrus. Alright. Now, how does polyembryony occur? Now, polyembryony can occur by various means, okay? Like simple polyembryony, for example, in gymnosperms. If you remember gymnosperms, so in that case you would know that in gymnosperms a single ovule can possess 2 to 8 archegonia, isn't it? So archegonia in the venter will possess one egg, right? So in that case, inside the ovule of the gymnosperm, naturally multiple eggs are present. And those multiple eggs after fertilization will give rise to multiple embryo, isn't it? So naturally a single ovule in the gymnosperm would possess multiple or polyembryo. So this type of polyembryonic condition developing would be termed as simple polyembryony. Okay. Next. Adventine polyembryony. Adventine polyembryony is common in citrus. Okay, like see this adventine embryony. What had I told you here? In the ovule, the cells of the nucellus or integument, okay, they can directly give rise to diploid embryo, right? So suppose I take in my example that this is the ovule and this being one of the cell of the nucellus which is diploid and has given rise to diploid embryo, right? And this is one cell of the integument which has also given rise to diploid embryo, okay? And naturally also there is an egg present, right? So when fertilization process will take place, this egg will fuse with a male gamete and give rise to a zygote. And the zygote will give rise to an embryo again, right? So when this ovule will convert into a seed, it will have three types of embryo. One is nucellar embryo, one is integumentary embryo, and another is bisexual reproduction, the zygotic embryo, isn't it? So already this ovule, when converts into a seed, it will have nucellar embryo, integumentary embryo and zygotic embryo. So this type of polyembryonic condition development will be known as adventine polyembryony. and this is again very common in citrus. Alright, so I hope apomixis and polyembryony are clear to you. Now you need to also know what is the importance or application of apomixis. Okay, let's see. So now regarding the importance of apomixis, first of all, hybrids with superior characters can be introduced with apomixis. See, in this chapter itself we have learned a technique called artificial hybridization, isn't it? In this method, what we were trying to do, we were trying to breed two plants to bring about their superior characters in a single plant, in a single hybrid, isn't it? So hybrids are always preferred to grow many cereals or crops because they have higher yield or higher productivity. Okay, that can be more beneficial to the farmers or agriculturists. But then there is a drawback of hybrid seeds. If you take seeds from these hybrid plants and you sow them, in the next season, those superior characters or those combination of characters which were there in the hybrid, that might not be seen in the progeny. So that might get lost. Okay, suppose there was a hybrid, disease resistant and higher in yield. Okay, now you make this particular hybrid seed grow into a plant. So now the plant that will be formed, it will also bear seeds the next generation. These seeds, they might not carry the same combination of traits in them. Means these seeds may not be disease resistant or higher in yield. Alright, so usually in hybrid seeds, this is the problem, that they cannot maintain the combination of characters within themselves, and usually the characters are lost over the generations. So if we can introduce certain apomictic genes in the hybrid seeds, so what will happen? They will always generate seeds by apomixis. So apomixis is after all a process of asexual reproduction, isn't it? So it will not show any genetic recombination. It will give rise to clones. So in that case, every year producing hybrid seeds won't be necessary, and that will be economically very beneficial to the farmers. Alright. Next, embryo formed by apomixis are usually free from infections. So this being the importance of apomixis, and with that we come to an end of this chapter. So sexual reproduction in flowering plant chapter being completed here. I hope all of you have been clear about the chapter and have liked it too. So in that case, the next class will start a new chapter, that is strategies for enhancement in fruit production. This chapter again has been divided into two parts. One part is under zoology, that is animal husbandry. Another part is under botany, that is plant breeding. So we'll start in the next class, plant breeding. Thank you.